Bueno, buenas noches a todos, buenas tardes para México, Bogotá, para los que están encima nuestro. Es un placer enorme, enorme darles la bienvenida a este nuevo ciclo de webinars a cargo de la CID, especialmente a cargo del Comité Educacional, eh, que se ha encargado con mucho trabajo de organizar este ciclo que, como les digo, eh, va a tener una frecuencia mensual, así que nos vamos a ver muy seguido, por suerte, y también quiero agradecer al Comité Junior porque van a participar activamente de estos webinars. Esperamos, la verdad es que esperamos aprender juntos, compartir experiencias, fortalecer nuestra sociedad y quiero agradecer nuevamente a los comités, darles la bienvenida de nuevo, agradecer a Carolina, a Luisa de Matos y a Belén Almejún, que es una invitada de lujo, una gran profesional, una gran persona, así que es un honor contar con ella como primera disertante de este nuevo ciclo 2025 de webinars de la CID. Así que muchas gracias a ustedes, Carolina.
I'm going to speak in English. So
Yeah.
I'd like to thank for everyone who's joining us here. A special thanks to Dr. Ricardo Sorensen. I saw your name there, Dr. Ricardo. It's a great honor to have you here. So if you want to say a few words here, we would be very honored to have you saying something to us here. And I would like to thank to all the members from the Educational Committee, Belén, Astrid, Juan, Alejandra King. We have been working to prepare these webinars, monthly webinars for you, together with the junior committees. We'll be having like the speaker and a case report that will bring us the daily application from the topic that we just discussed. And before we start with Belén and Luisa, I'd like to invite you for the next webinar. It will be in March 26, and we're going to talk about CAR T cells in primary immunodeficiency. So Belén, I'm pretty sure most of you know her. She's a Bachelor of Biological Sciences. She's a doctor in Biological Sciences. She has a specialty in molecular biology, immunology, and her research is focused in search, modeling, and functional studies of novel genetic variants derived from exome analysis of pediatric patients with congenital errors of immunity. And Luisa, she's from the junior group from LASID. She's a biomedical scientist from Brazil. She works in my group. She's just finishing her master program. And she will present a case about how to work with VUS variants. So Belén, you are very welcome to start your presentation. Thank you very much to all of you.
Thank you, Carolina. I will share my presentation. Let me know if you can see.
Yes.
Yeah.
Okay. Okay. Well, good afternoon or good night, everyone. I'm very happy to be here in this first webinar of LASID 2025. And as Carolina said, I will talk about VUS functional study for VUS, taking into account our experience in the diagnosis of inborn immunity in Argentina. So, as many of you know, but I think that some student of my faculty is already connected, and I introduced that what are inborn of immunity for the student. As you know, are a broad set of monogenic disease that affect various aspects of the immune system, compromising its ability to fight infection. And also individual rare, together they represent a significant burden of the healthcare system. These conditions are often underdiagnosed because their signs and symptoms overlap with a wide range of genetic defect in different genes. For many patients with an inborn of immunity, conventional clinical diagnosis can be insufficient. In fact, severe case may lead to clinical complication or even death before the underlying molecular disorder is identified. So this is why the genetic testing, particularly whole exome sequencing, can make significant differences in the quality of life of the patient if the causal gene is found. Identify the responsible gene is not only vital for clinical diagnosis and precision medicine, but also enhance our understanding of the complex mechanism with no redundant pathway essential for the development and homeostasis of the human immune system. So VUS is a powerful tool for identifying genetic mutation that may cause disease and hereditary disorders. And this technique involves sequencing all protein coding regions of the genome, representing 1 or 2% of the entire genome, where most disease-causing mutations are found. So it includes only protein coding region. This is a remarkable thing, but because not all exon, because some exon or exon part function as a regulatory zones without coding properties. They do not translate into proteins. So we conduct VUS in our country on samples from patients with inborn of immunity across the country, thanks to a collaborative effort involving hospital and clinic associated with AINCA, the association, I will say in Spanish, la Asociación de Inmunólogos Clínicos Argentinos. And between 2023 and 2024, we analyzed 200 samples, 235 for more than 200 samples for I not be exact the exact number. This year we could have processed more, but we have a currency devaluation at the budget freeze in science and technology by Milei's government due to more than 30 samples are currently awaiting VUS processing by the research laboratory, our research laboratory. So I will tell you how we work. Here's what we do in the lab from the patient sample to generating the genetic report. We receive the sample, the blood sample, anonymize it, and assign a unique code. We extract the DNA, store part of it for future study, and send part to the sequencing companies like Celemix, Macrogen, Trivillion, or Novogene. And these companies fragment the DNA, capture protein coding region, purify those fragments, and sequence them. It is important to note that while we have sequencing facilities in Argentina, the costs are higher due to the recent imports, making outsourcing more economical. So nevertheless, having national result will be ideal to bridge this gap. The company returned to us 100 of reads, which need analysis from bioinformatic pipelines that we do in our laboratory, thanks to the collaboration with all the bioinformaticians in the Faculty of Exact and Natural Science. So essentially, the reads are aligning to the reference genome, covering DNA region as shown here. Variants are colored for region differing for the reference genome, created a variant calling file or VCF. The variants are then annotated by chromosomal position and change, and we upload this case to Argentinian platform named Vicina B platform that allow us to prioritize or do the prioritization of the variant in the exome of our patients. This is what the bioinformatics do. They do a lot of stuff to do more easily for us to do the variant prioritization. So this prioritization considers various factors, whether the variant is synonymous without no amino acid change, or missense causing an amino acid change that impact in the function, may impact in the function of the protein, or a friendship due to insertion or deletion that leading to a completely altered protein beyond that point, or may this insertion may cause an unstabilized mRNA that go to degradation. Finally, we have non-friendship mutation that that is insertion of deletion. There are multiple 3 bases that insert a specific amino acid and could lead to may impact the function of the protein if these amino acids are in some part that are necessary to the functionality of that protein. Finally, we have a nonsense mutation to introduce an premature stop codon, and this mutation may result in a truncated protein or in an instability of the mRNA that go to nonsense-mediated decay. We also, the mutation can also affect the splicing site by altering mRNA and potentially deleting intronic or exonic segments. And all these changes are evaluating using conservation, structural, and physicochemical predictor, as well as meta predictor that suggests whether a change may be harmful. So variant interpretation also depends on population database comparison. Variants not associated with disease are commonly in the general population, whereas pathogenic variants are rare. We rely on databases like Genome ID with over 1.5 million alleles. However, we have limitations remain due to the underrepresentation of the American population, impacting in our genetic diversity reference. But if we have more variant, it's more easy diagnosis rare disease. And if we have more variant in the databases, we can better understand the genome function. Additionally, for variant interpretation, we verify finding using Sanger sequencing, the gold standard technique, ensuring the variants presence in the patient and familial segregation to check inheritance patterns, heterozygous and homozygous pattern. The family segregation helps to determine if the variant co-segregate with the disease by recessive inheritance as homozygous component heterozygous, or by dominant, often de novo mutation, or X-linked recessive with healthy female carrier and affected males. Using all this data, we carry out variant labeling according to the guideline and standard of the American College of Medical Genetics, ACMG, and based on the impact of the change, population frequency. This is the impact, the population frequency. We accumulate the pathogenicity levels, the family segregation, and we determine if the variant is pathogenic, likely pathogenic, VUS, variant of uncertain significance, likely benign, or benign. This process allows us to create a genomic report. But it's important to know that previous to the genomic report, the report involved not only the filtering and prioritization provided by the platform, but also continuous collaboration and meeting with physicians, geneticists, and molecular biologists to assess whether a candidate variant matches the patient's clinical presentation. Only after this collaboration is the genomic report prepared. In cases where the VUS results are negative, we check the coverage of immune-related genes because some genes are not well covered by VUS on some exon of some genes, and consider a possible popping variation or CMVs, and save the data to future analysis, since our current limitation may be due to a lack of knowledge. So often, a genetic report results in a VUS or variant of uncertain significance. For example, in this chart, I illustrate the percentage of variant identified in a patient's VUS, where more than 90% are benign or likely benign. A minimal percentage are pathogenic or likely pathogenic without implying causation or relation to the specific pathology, and around 9% are VUS. And not all VUS are the same pathogenic potential. Some may be more pathogenic than others by the labeling of ACMG, as I told you recently. And while most are found in genes with limited association with the clinical phenotype and should not be reported, others may hold clinical relevance, requiring thorough evaluation, and could be the step toward identifying new gene function and pathways of the immune system. So a VUS is often classified as such due to absence of functional tests confirming or refuting its pathogenicity, inconsistent annotation in databases, or unknown segregation patterns. The more information we generate, the more we can reclassify this variant and better understanding those are genuinely pathogenic. So to determine the pathogenicity of a VUS, functional assays are frequently needed, particularly in the context of inborn of immunity, where allelic and locus heterogeneity complicates determining causality. When one gene causes many phenotypes or one phenotype being caused by many genes. So this test can be conducted on the patient sample, stimulated using expression plasmid or through the gene editing, and they can be in vitro or in vivo. Basic tests may involve determining protein expression levels, where a lack of expression will add a pathogenicity level to reclassify the VUS as a pathogenic. Different conditions may require specific analysis for functional validation. So this test can be conducted on patient sample, stimulated, this is different disorder will have different assays for functional validation, no? This test can be conducted on the patient sample, such as by gene editing. And we also work with healthy donor cells and patient cells, measuring RNA or protein expression. So if we can measure an RNA expression or protein expression in healthy cells or patient cells, we can add another labeling to the pathogenicity of the variant and reclassify the VUS as a pathogenic. We also, the functional tests involved cell stimulation and activity measurement are also possible, but different cells have distinct function. This is an example. If we have a mutated gene that may be involved, but we think that may be involved in the B cell development, we can stimulate in B cells, and as an example, no, and measure the levels of immunoglobulin in the healthy control and the patient sample, and see the activity of that cell in vitro. As I say, different cells have different function and need different stimulation, so we need different in vitro assay for each mutated gene. So this process involves skilled personnel, and it's essential for definitive diagnosis. As mentioned at the start, over the past 2 years, we have analyzed more than 200 samples nationwide. And during the first year, most samples came from Buenos Aires City, but this year we expanded to include Buenos Aires Province, Santa Fe, Córdoba, Mendoza, and Tucumán, where training professionals for diagnosis and treating inborn of immunity are available. The gender distribution was balanced between male and female patients. And regarding the type of inborn of immunity diagnosis, also predominantly antibody deficiency are more represented. All categories of inborn of immunity were included. For the variant prioritization, we use as I said, the Argentine Virginia platform in collaboration with the group of Dr. Marcelo Martí and the technician Jonathan Zayat that made this possible. And we don't analyze all the genes in the genome, so we only focus on a subset of genes selected by our team. Specifically, we analyzed the 485 genes, US genes associated with inborn of immunity. This changed in the last months, and we also analyzed the last other gene by the US. And also we have 452 non-US genes associated with immunopathway. Some of them nowadays are US genes related to inborn of immunity. And we also analyzed 108 epigenetic genes, a mutation in which have shown immune system issues, and 696 genes related to the nervous system due to a patient with syndromic combined immunodeficiency, often presenting with neurodevelopmental features. Nearly 50% of the sample were positive for a genetic diagnosis. This positivity is bigger right now because we reanalyze with the novel VUS genes, and some negative sample go to the positive for our fortune, will go to the positive patient's diagnosis. So in the positive sample, we have 83 VUS genes and 30 non-VUS genes until November. From these 30 non-VUS genes, 10 were cataloged by the US associated with involvement of immunity, 1 epigenetic gene, and 4 neurogenes. And in those patients, we also have a mutation, another mutation, a second hit in 5 neurogene and 5 immune non-VUS genes also. We have a 34 sample in progress because these are under review due to an ongoing discussion, monoallelic finding in recessive genes, or involvement of non-categorized genes require extensive validations. And we observed the distribution of positive variant by their effect type and inheritance mode, with a prevalence of missense variant and dominant inheritance. And this prioritization and labeling process according to ACMG led to 26% of the variant being classified as pathogenics and nearly 60% as a VUS. This is a work that is going by Lorenzo Herrera, is part of his doctoral thesis, and we are in the process of writing this work. We hope that in the next months we have a publication of this result of our cohort. So let me share some representative cases of the variant identified in our patient population. As an example, in 2 brothers treated in the Clinical Immunology Center in Buenos Aires by Dr. Moreira, who exhibit similar immunological profile with recurrent infection and neutropenia, we found a French mutation leading to a premature stop codon and resulting in nonsense-mediated decay degradation of MACT1 protein. These 2 brothers have the same mutation, but they have different phenotype. As you know, this protein plays a fundamental role in intracellular magnesium function, which is essential for T-cell receptor activation dependent on magnesium influx. And the gene is located on the X chromosome, and mutation in gene lead to Exome syndrome characterized by CD4 lymphopenia, chronic infection, and EBV relating in lymphoproliferative disorders. One of the brothers presented EBV-induced HLH and developmental delay. And considering that MACT1's role in neurodevelopmental development is still debated, we checked whether the brother with developmental delay had any additional variant in the neuropanel. And we found a pathogenic variant that was presented in him, but not in his brother. Both are novel. This mutation is novel in the brother, and potentially explain the genetic differences between them. Other double heterozygous missense variants were found in CARMIL2 gene in a patient treated by Dr. Uriarte at the Intersonal Maternal and Child Hospital in Mar del Plata. This patient presented with CD4 lymphopenia, impaired T-cell proliferation, autoimmunity, and recurrent infection, indicating combined immunodeficiency. As you know, mutation in CARMIL2 specifically affects T-cells, and the protein is crucial for cytoskeletal reorganization. The identified variant has extremely low frequency in the healthy population and were classified as VUS. And this is plausible for a gene with recessive inheritance, and one was inherited from the mother. We do the segregation. We don't have sample from the father because the father is absent, but the mother only presents one of the variants, so we think that the other is from the side of the father or is the novel mutation. So the patient clinical presentation matched with the previous single report describing 3 patients with HPV-related conditions. Another case involved a patient from Mendoza, monitored from the Fleming Children's Hospital by Dr. Emma Nievas, who had a mutation in SP1 gene. And this is a particular case because the patient was agammaglobulinemic, such as the other 6 patients that was reported until November, December. But the patient of Dr. Elma Nievas presented B-cell in periphery. So the mutation found led to a frame and an early stop codon may result in nonsense-mediated decay. And nowadays we are testing if in the mRNA of the cell from the patient and her mother who present the same mutation is only expressed the healthy allele. And this also is particular because the mother presents some clinical presentation, but she has hypergammaglobulinemia. So this mutation is an interesting example that the clinical spectrum of PUMA is not fully understood nowadays. So this case further broadened the clinical spectrum associated with SP1 mutation, providing additional insight into PUMA manifestation. So another example is a patient treated by the Duran Hospital by Dr. Lopez and Paulini, who had bilateral hearing loss, frequent infection, autoimmunity, and hypogammaglobulinemia. And in this case, we found 2 variants in the LRIV-A gene that segregate correctly. Both variants were absent in the healthy population databases and affected splicing or induced friendships, leading to nonsense-mediated decay. Broadening the clinical spectrum, recessive LRV mutation, because the LRV are essential for vesicular trafficking and recycling CTLA4 to the T-cell membrane. I also want to mention case where genetic finding guide treatment decision. One such case involved a child from Córdoba Municipal Hospital treated by Dr. Sasia, in whom we identify a mutation in the SH2D1A gene, which is X-linked and causes X-linked lymphoproliferative syndrome. The treatment for this condition, as you know, is a bone marrow transplantation, and family segregation analysis performed at Garand Hospital confirmed that only the mother was carrier of the variant and none of the patient's siblings were carriers, enabling comprehensive genetic counseling for the family. This child was have a bone marrow transplant in November of 2024. Another example is a patient with an homozygous variant in the 2 gene, ADA2, monitoring by Dr. Lucia Peirano at the Cruce Hospital. And the treatment option include enzyme replacement therapy or bone marrow transplant. And additionally, there is a potential for treatment with a JAK inhibitor as demonstrated in a patient treated by the Maternal and Child Hospital of Mar del Plata by Dr. Uriarte. This patient had a likely pathogenic variant in the STAT1 gene, matching with the clinical presentation of a STAT1 gene gain of function condition. So I am boring you. Maybe we have 25 minutes of talk. So now I will tell you about some functional assay that we have developed in the laboratory of over the last few years for the VUS variant that, as you know, is the major percentage of variant that we found in WES. The first we conducted involved a functional assay in a scaffold protein that ultimately activate the transcription factor NF-kappa B in both B and T lymphocyte. Mutation in the CARD11 gene must be functionally evaluated because they result in multiple clinical phenotypes and their phenotype overlap with mutation in other immune system genes. So Lorenzo Herrera and Julieta Fernandez put this assay in our laboratory, set up this assay. I don't have the correct word. Set up this assay in the laboratory. So we transfected cell lacking CARD11 with a novel variant that we we make by mutagenesis and activate the cells to assess functionality using a reporter gene assay, while also evaluating their expression and cellular distribution. So this assay allow us to determine that one of these VUS variant did not cause pathogenicity. So we label this variant as a benign, and 4 showed gain of function and 4 showed loss of function activity. Simultaneously, we evaluate novel mutation in the syntaxin 11 and BC11B genes presented in a patient with the VUS syndrome, thus the syndrome and developmental delay. This patient is treated by Dr. Mariana Vila at the Garrahan Hospital. As you know, syntaxin 11 is a snare protein necessary for the degranulation of cytotoxic granules and in T and NK cells, while BC11B is a transcription factor required for T cell and neuronal maturation and differentiation. So, Ana Colado, Mercedes Borges, and Lorenzo Erra, using functional assay for cytotoxicity, protein, and RNA expression, they perform the assays in our laboratory in the patient and healthy donor cell, as well as they do the secretion of interleukin-2. And we observe aberrant functionality in our patient, potentially influenced by both mutations. Notably, the syntaxin 11 gene variant segregates with autoimmunity in the family, and BC11B is also present in the mother who does not have a neurological defect. This work was recently publicated in Eligen. So if you want to read it, it was approved at the 10th of February in this year. So another assay developed in the laboratory by the biochemist Diana Flores, but the magister, right, the Nobel Magister Diana Flores, and now continued by the student Mora Bertoni, involved STAT3 functionality, as mutation in this gene result in diverse clinical phenotype and overlap with mutation in other immune system genes. So we have a lot of variant in highly conservative position, all VUS, and by the bioinformatic predictors, almost love variant, almost all the presumable love variant predicts the pathogenic, but the gain of function, the possible gain of function variant result in ambiguous result. And also one variant that it was in a patient with a phenotype that was made due by a love variant was classified as uncertain significant for all the predictors, the bioinformatic predictors. So Diana evaluates functionality by doing simulated the mutation in a plasmid and affected cell mission STAT3 reporter gene after cytokine stimulation and assessing the phosphorylation of this transcription factor in various mutants. And this both assay allow us to determine that the loss of function variant were lost, but the gain of function variant in our hand in the laboratory exhibit GOF feature in the in vitro assay, highlighting the importance of performing this kind of assay to study variant not well predicted by informatic tools, and also not well predicted with the self of patient by flow cytometry when the cells of the patient are stimulated and measure the phosphorylation of this transcription factor. And this assay allows us also to establish that the mutation in the position K707N exhibit a remarkable loss of function profile in the in vitro assay. So We have also established a assay to measure the functionality of PTPN2, a phosphatase that inhibits STAT3 pathway activation. The student Nicole de Graaf did the assay to test the functionality of two variants representing a patient monitored by Dr. Oleastro, one inherited by the maternally and the other paternally. The findings suggest that the novel PTPN2 variant in the laboratory contribute to the dysregulation of STAT3 mediated signaling pathway, and also we performed the STAT1 phosphorylation assay and STAT1 activated functional assay, and we observed the same pattern of loss of function of the PTPN2 phosphatase, leading to that allow us to say that both mutation may lead to the polyautoimmunity and pro-inflammation that we observe in the patient. Finally, I want to share an interesting case involving interleukin-10 receptor alpha, which has an unusual history. It began with Silvia Daneyan mentioning the case of a girl treated at the Gutierrez Hospital by Dr. Florencia Biasoli, who had an inflammatory bowel disease-like clinical presentation and an heterozygous mutation in interleukin-10 receptor alpha found on a gene panel. As you know, homozygous mutation in interleukin-10 receptor alpha causes IBD and requires bone marrow transplantation, but this mutation creates a premature stop codon that eliminates the entire intracellular region of the protein, and is located in the exon 6 at the position -21, and then in the mRNA who may escape to the nonsense mediated decay mechanism. So months later, a VUS of a patient with CVID, combined immunodeficiency and lymphoproliferation, followed by the Hospital Muñiz by Dr. Carla Chaclevicius, revealed the same variant. So discussing this with Carla, we learned that he was the cousin of the girl's mother, and that another child followed at the Hospital Alemán also had inflammatory bowel disease, with multiple family members showing an autoimmune manifestation. So this suggests a possible case of dominant inheritance with a variable clinical presentation and penetrance. So we collaborate with Soledad Gori, and together with Lorenzo, we're finding that the heterozygous variant blocked STAT3 activation when the cells of the patient were stimulated with interleukin-10. And also, site-direct mutagenesis was employed to introduce the variant into the interleukin-10 receptor alpha wild-type sequence, and we transfected 293 T cells with the plasmid containing wild-type or mutated sequence, together with the STAT3 GFP reported plasmid. The STAT3 transcriptional activity was measured, and as you know, you may see here, the mutated allele blocked the GFP expression by STAT3 transcriptional factor. So our findings suggest that this heterozygous interleukin-10 receptor alpha variant acts via dominant negative mechanism, truncating the receptor cytoplasmic domain and leading to the variable immune phenotypes and penetrance. So this study highlights the possibility to consider autosomal dominant inheritance in some interleukin-10 receptor alpha-associated immune dysregulation. In summary, with everything we presented, or I presented here, it's clear that VUS has proven to be an invaluable tool for diagnosing inborn immunity, revealing the genetic basis of many cases that would remain underdiagnosed by conventional methods. This study had a direct impact on patient treatment, including therapies such as transplantation, enzyme replacement, or JAK inhibitors. Moreover, this work reduced the burden of the healthcare system. However, the process is not without challenges. Approximately 60% of the variants we identified were classified as variants of uncertain significance, requiring further study. So this VUS pose a dilemma for clinicians, as determining their disease causality require additional functional study or family segregation analysis. This underscores the need for ongoing research and collaboration between geneticists, immunologists, and bioinformaticians to better interpret this finding. And looking ahead, we have several key developmental areas. We want to expand in the use of the whole genome sequencing to capture a broader range of genetic variants, including non-coding region, structural variant, and copy number variation that may contribute to the disease phenotype. And also, the functional validation of VUS remains a priority, and we plan to continue in vitro study to clarify the pathogenic role of the variant. And we need to establish international collaboration. This partnership will be essential for validating the novel genetic finding and enriching global databases, ultimately enriching the interpretation of rare variants. This work is not only possible thanks to the collaboration and commitment of all the institutions and professionals dedicated to studying inborn of immunity, and of course the patient and their families, and INCA that do this possible too. Well, lastly, I invite to support our need, especially for adult patients, for confirming and segregated identify variant via Sanger sequencing to enhance the diagnosis of rare disease in Argentina. We have launched a donation campaign through the Faculty of Exact and Natural Science Foundation to fund the purchase of Sanger sequencer to amplify diagnosis and variant confirmation for this and other conditions. Lastly, I cannot end, unfortunately, without noting in this context of an international talk that public education, which I am product of, public health, where most patients are treated, and the scientific and technological system as a whole are under attack by our government, which sees us as an adversaries. Skilled individuals see no future due to underfunding, program closure, and low salaries. We stand in defense and need the work to know our reality, because education, health, and public science in Argentina are also a cornerstone for Latin America. I call on everyone to defend these pillars, because we cannot allow the light of our future to be extinguished. So thank you very much for listening to me. I expect no bother you. If you have any question, I listen. I don't know if the question is right now or at the end of the talk. Let me know, Carolina.
So thank you very much, Belen. I think it was really interesting, your talk, with all the examples that you gave us. And I think it's a real challenge for us, right? The VUS are a real challenge for us, because it's really like for us to establish a diagnosis and maybe to refer the patient for a bone marrow transplantation, for example. If we have a VUS, it's tricky, it can be dangerous, right? So it's really important that we talk about this. And we know that it's not easy to get access to genetic study in many places in Latin America. And now we have this new challenge that is the VUS. So it's very important that we collaborate, because we cannot do all the things all the time. But if we establish a network of collaboration, we can do a lot of things for our patients. So thank you very much. If you have any questions, you can write down on the chat. So Alejandra King already said that it was an excellent presentation, muy clara. And if all the people that are watching the webinar, if you have some questions, so please write down in the chat. And after Luisa's presentation, we can go back to the questions. Is that okay like this, Belen?
Okay, perfect.
Okay, okay, great. Yeah, so now we have Dr. Luisa's talk. Luisa is from the Lazid Junior from Lazid. And the model.
Carolina,
I think she lost connection.
okay.
I will text her. Just a second.
Maybe we should continue.
Okay.
Thank you, Dr. Luisa. Would you like? Oh, here is Dr. Carolina.
Oh, I'm back. Okay.
Okay.
So I don't know how much you listened to what I was talking, but basically when I realized that I was not with you anymore, I was saying that Luisa, you could start your presentation.
Okay. Okay, can you hear me well?
Yes, doctor.
Okay. So good evening. My name is Luisa de Matos. I'm a biomedical scientist, as Dr. Carolina already said. And I'm currently working on my master's degree. And the case that I will be presenting today is a case that happened in our project and that we managed to reclassify the variant as likely pathogenic using the functional testing. So let's start talking about the clinical case. So our patient was a female, full-term birth, normal weight, with not reported consanguinity in the family. But she had a history of sister death at 5 months old due to infections. And she also had 2 other healthy brothers. So her first clinical manifestation occurred when she was 5 days old, with lesions on the oral cavity and also in the perineum. And those lesions were associated with high fever. So she was admitted to the ICU on 2 occasions by that date, when she was 5 and when she was 15 days old, due to the same clinical symptoms. Around that date, she also presented with 2 pneumonias. So after a while, when she was 6 months old, she was admitted to the hospital due to disseminated herpes and also herpes otitis and urinary tract infection. So after a month, she was admitted again to the hospital due to abdominal pain, food refusal, and vaginal and anal lesions. Those lesions did not improve despite the treatment. So with that clinical manifestations, it was required that we perform the genetic testing. So we performed the exome sequencing, and with the test, we identified a variant in the interleucin-10 receptor alpha gene. This variant was in homozygosis. At first, the only criteria we could apply was the PM2, because the variant has extremely low frequency in population databases. So our next step to evaluate this variant was to perform the family segregation. So here are the results of the family segregation. Here we can see that our patient was the only homozygotic the only one in the family that carries the variant in homozygosis. Every other member of the family was heterozygotic. So with this information, we could apply 2 more criteria. The first one was the PP4, because the patient's phenotype was highly specific for the disease we were studying. And the second one was the PP1, because as I said, the patient was the only one in the family that was homozygotic homozygotic for the variant, and also the only one in the family with the symptoms, as far as we know, because we couldn't test her older sister, because by the time that we tested the family, this older sister had already passed, so we couldn't test her. But even with all this classification criteria, we were still dealing with a VUS. So we had to go back to the basics and study what we could do to change this classification. So the deficiency of the alpha receptor for interleucin-10 causes very early onset inflammatory bowel disease. And this condition will cause severe gastrointestinal symptoms such as fistulas, perineal abscesses, and also colitis. This happens because the interleucin-10 is very important for immune regulation, right? So acting mainly to inhibit the pro-inflammatory activity, and also because this cytokine is produced by a great variety of immune cells. So it happens by the binding of the interleucin-10 to these receptors, the alpha and the beta receptor, that leads to intercellular signaling pathway, leading the JAK1 and the TUK2 mediated phosphorylation of the STAT3. So the STAT3 dimer that was generated by this interaction undergoes nuclear translocation that induces the activation of a specific gene encoding for anti-inflammatory factors. So the inflammatory response is regulated. With this information, we decided to evaluate the STAT3 phosphorylation in vitro through the interleucin-10 stimulation. So we could study the receptor function. To start, we isolate the PBMC probands because those were the cells that we were interested in studying, following the phosphorylation assay. For this assay, we divided the cells in 3 groups based on the stimuli by the EL10 and also the antibody labeling. So we could compare the response with the spontaneous one without the stimulation and with the stimulated one with the simulation, sorry. And finally, we could detect the phosphorylated stat by flow cytometry. And then we have the results. Here you can say that these numbers represent the measurement of the fluorescence by the flow cytometry. And here we can compare the spontaneous values to the stimulated values. When comparing to the control, we can check that the proband's fluorescence ratio represents a very lower fluorescence ratio, right? And with this information, we can say that the variant causes impact on the cellular response. We also tested the mother of the proband that was a heterozygotic, that has the variant in heterozygosis. But the mother, the test of the mother showed the very similar fluorescence as the control. So we can say that in this case, the variant in heterozygosis didn't cause the clinical phenotype as the homozygosis variation. And here we have the previously applied criteria, the 3 previously applied criteria, and we could add 1 more criteria because the functional studies show the damaging effect on the gene or the gene product. And according to the American College of Medical Genetics, when you have 1 strong criteria plus 2 supporting ones, you can reclassify the variant as likely pathogenic, and that was the case. So with this functional testing, we could conclude the case and conclude the diagnosis of this patient of ours. And that's it. I want to thank you for your attention. I want to thank you also Dr. Carolina for her advisor and Labhara's team, that is our lab group. and also Dr. Jesmar for sharing the protocol. Thank you.
So thank you, Luisa, for your presentation. I'd like to tell you that the child is doing very well now after a bone marrow transplantation. So we are really happy that we could help this family. So anyone has any questions to Dr. Belén or Dr. Luisa? I'd like to remind you that the webinar will be recorded in the LASID website, so you can watch this at any time. So it doesn't seem, so everyone understood everything, so we don't have any questions. Belén, would you like to make any comments, any additional comments?
No, I only I am d'accord in French. I with you with your talk and your words, we need to collaborate between different laboratory of Latin America. We need to join and to be more. I think that we are more strong together. We have some particular economical feature in a lot of a lot of country of Latin America, and we have very professional that are extremely, I don't know how to say, compromising with the patient and their diagnosis. So if we have some strong in the laboratory in Argentina or in Brazil, we need to or in Chile, we need to collaborate with another country, and also to present some financial of international grants to do a better diagnosis, a better understanding of the involvement of immunity in our particular population, and also to have a increase the databases of our genetics basis. I think that we have a problem with the alleles that are in the genomic database of the worldwide genomic database, because we have a lot of presence of Europe alleles, and the American mixed genetic background is not present, and so some variant that we classify as VUS may be present in our population, and we don't see it. And this is a challenge that we have as a region at all, completely the complete region of Latin America. So I think that these talks, this webinar, may strong the I don't know how to say, I will say in Spanish, fortalecer los lazos que tenemos entre los distintos países, como para que podamos hacer más fuerte a la CID y más fuerte la investigación y el diagnóstico en Latinoamérica. Sorry for saying in Spanish, but sometimes when I think with my heart, it's difficult to find the words to say.
Yes. Yes, and Alejandra said that she cannot talk because it's very noisy, the place where she is, but she agrees, and she says that we have to start working together as soon as possible. So now we have a few announcements for you, and Alfonso, you can share the announcements here for us. And so you still have a few minutes to write your questions or comments if you have some of them. So we would like to invite you for the Brazilian Group of Primary Immunodeficiency meeting. We have the 2025 meeting in São Paulo. It will happen in April 25 and 26. So we have already, the registration is open, and you can find us on Instagram webpage or the website, the Bragid website, and it would be great to have you here in São Paulo with us. Also, together with the Bragid meeting, we're going to have the GEBRAE. It's the Brazilian group for hereditary angioedema. So this one will be on April 25, and then we have Bragid 25 and 26. So they will be in the same place. And next one, very important, please save the date. We have our biannual LASID meeting in October, and this year the meeting will happen in Buenos Aires. So we hope to see you all there. And also join us to LASID, so you can just point your cell phone camera to the QR codes that you have here, or you can go to the lasid.org website. And you can also join us as a regular member or as a junior if you are under the age of 35, or if you can prove that you are still in immunology training. And we need you to be part of LASID, because the more we can put together immunologists and people interested in immunodeficiency in Latin America, the strongest we'll be. And this is very important for us to develop our science, to develop care for our patients, and to bring more and more benefits for them. So as we don't have any questions, we'll be finishing our webinar now. And once more, I would like to invite you for the next one. It will be in March 26, the same hours that we have this one. And we're going to talk about CAR T cells. So thank you very much.
There's a question, sorry.
Okay, question where? Where? Oh, I found it here. So we have two questions. So, Ricardo, thank you for being here. We tried to invite you to join us and maybe say some words here. Ricardo says that there are some of these mutations associated with specific infections so that they can manifest clinically late in life.
Okay, Ricardo, I'm sorry, but I am biologist. I don't see the patient, so I cannot to say if a specific infection is in some of this patient. But I have tarea for the next talk, and I can answer your question. Sorry, but not I think that is not a particular infection, but I cannot say affirming totally, because I don't see the patient. I only have an anonymized sample. So we talk with the physicians and the clinician and discuss the mutation in the context that they know all the clinical, we know all the genetics. So we have this compartment in Argentina that the research is in the laboratory, and the clinician did not do laboratory research. And this is some of our impossibility that we are right now with the red, I don't know how to say, with the AINCA. We collaborate with all the hospital to do this assay and better understand the pathophysiology of the immune, the inverse of immunity. So, sorry for...
No sé si me escuchan.
Sí.
Bueno, entonces realmente me emocionó verlas a todas, a las que conozco y a toda la nueva generación. Miren, Latinoamérica y la CIT tiene que trabajar como ustedes lo están haciendo. No esperen. Van a recibir ayuda de muchas partes, pero sí Latinoamérica tiene un potencial tremendo. fue la segunda sociedad de inmunodeficiencia en el mundo después de ESIT, y ustedes la están usando ahora, y ojalá trabajando también con el Journal of Human Immunity. Jean Laurent me prometió que les daría espacio, ojalá hasta para presentar preguntas, etcétera. Voy a tener que volver a conversar con él al respecto, pero la felicito, realmente fue un placer escucharla. Y bueno, yo estoy un poquito pasado de moda, ¿entiendes? De manera que mis preguntas no pueden estar al nivel de lo que ustedes están haciendo. Pero estas cosas son tan complicadas que si no colaboran, no se va a lograr avanzar mucho. Así es que muchísimas gracias y por presentar estaré en estos seminarios todo lo que pueda y ojalá también promoviendo otras actividades y ayudándole. Luis, es fantástico, realmente. Así es que muchas gracias y crean en ustedes, crean de creer en la capacidad de Latinoamérica y trabajen juntos, es tan importante. ¿Ya? Escuché que está Alejandra King también por ahí, creo, y en fin, y obviamente, así es que la felicito y fue un placer. Eso es lo que...
Thank you very much, Ricardo. It's as I told you before, it's an honor, really like honored for us to have you talking to us here today. Thank you very much. Thank you for like it's because of you that we have all this today. So we really thank you for all your efforts to make LASID to happen.
Bueno,
So we
Bueno, lo
also
mismo que Carolina, gracias Ricardo por las palabras y la verdad que sí, bueno, es importante, yo estuve en la fundación de la CID y que el grupo fundacional y la verdad que creo fervientemente en nuestro potencial y es algo que me mueve a seguir adelante más allá de las dificultades y creo que, bueno, que nada, que juntos podemos hacer muchas más cosas y que individualmente terminaremos perdiendo, digamos, ¿no? La región como un todo se necesita esa unidad que también se ve en la CID y en cada congreso de la CID a los que participé, con lo cual gracias por las palabras y por la presencia en la charla también.
sí, Belén, fue extraordinaria la charla de Belén, realmente. Las dos charlas, pero
see. And we don't have any more like questions, but we have a comment from Kate Sullivan. She says that excellent talks. So thank you, Kate, for being with us today. And also Alejandra says muchas gracias a todas. And Alejandra, this is a work that you belong to. So you were as part of the educational committee, you and Belen, we were, and Astrid, I think I saw Astrid here, and in the participant list. So it's great to be working with you all. And just before we finish here, I'd like to invite you also to the Congreso Mexicano de Inmunodeficiencias Primarias that will happen at the end of March, 27 and 29. So if you can join the Mexican Congress, I'm sure you will learn a lot. We'll have Kate Sullivan, Alexandra Freeman, Lisa Forbes, Matias Oleastro, and Jacinta giving for sure excellent talks there. So thank you very much to all of you. And I hope to see you next month, March 26, for the next webinar.
bye bye.
Bye-bye.